Molecular identification of inherited diseases. Models of disease etiology. So genetic would be inherited, environmental would be somatic, and multifactorial are polygenic and somatic. Family history of phenotype is illustrated on a pedigree diagram. In a pedigree, typically men are squares, women are circles, and impacted individuals are shaded in. If an impacted individual, or if any individual is crossed out, that means that they have passed away. And sometimes you'll see individuals that are partially shaded, and that means that they're heterozygote for the impacted disease. Now, just by looking at this pedigree, you can see that only men are impacted. Typically, when you see such a high rate of men impacted and almost no women impacted, this is a sex-linked type trait. It's probably Y-linked on the Y chromosome, but looking at this, sorry, I take that back. It cannot be Y-linked because all men would have to be impacted. It's likely X-linked. X-linked would mean that since men only inherit one copy of the X chromosome, it increases the likelihood that the X chromosome that they get is going to be one of the recessive types or one of the bad types of the gene. So then you can see a much higher prevalence in men than women. A practical example of this is color blindness. Typically, in color blindness, it's X-linked recessive disorder, and typically men are primarily impacted. Women can also have color blindness, but it's in men to a greater extent. So autosomal dominant, you're going to see a lot of impacted individuals. Autosomal recessive would mean that you're not going to see a lot of impacted individuals. And sex-linked or X-linked recessive, you would see a lot of impacted males and not a lot of impacted females. You can see these ones are shaded in partially because those are carrier females. We can't have carrier males on an X-linked disease because males only get one X. Transmission patterns, AR or AD, so autosomal recessive, autosomal dominant, or sex-linked patterns are observed in a single gene disorders. Diseases caused by one genetic mutation. Prediction of a transmission pattern assumes Mendelian inheritance of the mutant allele. Transmission patterns, we can have gain of function, where mutations are usually display a dominant phenotype. Loss of function, where mutations usually display a recessive phenotype. Dominant negative, patterns are observed with a loss of function in multimeric proteins. Autosomal recessive is the most frequently observed transmissible pattern. The mutant phenotype is not observed in the heterozygous normal mutant state. A mutation must be homozygous mutant to show the abnormal phenotype. AR mutations can also result in an abnormal phenotype in a hemizygous mutant and deletion state. The loss of the normal allele reveals the mutant allele, and that's called loss of heterozygosity, or LOH. LOH results from somatic, environmental, not inherited, mutations or deletions of the normal allele. Hemochromatosis 1, overabsorption of iron from food caused by mutations in the gene for a membrane iron transporter protein, HFE. Thrombophilic state caused by the lead-in mutation in the gene for the coagulation factor 5, F5, and or specific mutations in the gene for coagulation factor 2, F2. HFE C282Y can be detected by polymerase chain reaction, restriction fragment length polymorphism. Detection of factor 5 latent, mutation by PCR RFLP, and mutation by SSPP. So sequence-specific primer can also detect it. Few diseases have simple transmission patterns due to variable expressivity, the range of phenotypes from the same genetic mutation. This is where I've talked about previously, where just because a gene is present doesn't mean that it's going to be expressed. And if it is expressed, it may not be expressed to the full extent. And if it is expressed to the full extent, it may not produce enough active product to be able to confer the disorder that we're testing for. Variable expressivity is talking about the range of phenotypes from the same genetic mutation. So, if a mutation is present, it may have a significant impact or a moderate impact or no impact at all, depending on the individual. Genetic heterogeneity, different mutations cause the same phenotype, often observed in diseases with multiple genetic components. Incomplete penetrance, the presence of the mutation but no abnormal phenotype. That's where the gene's present, but we don't see an abnormal phenotype. Single gene disorders, or disorders with multiple genetic components with non-classical patterns of transmission. Gonadal mosaicism, somatic mutation in germline cells. Genomic imprinting, nucleotide or histone modifications that do not change the DNA sequence. Nucleotide repeat expansion, increased allele sizes disrupt gene function. Mitochondrial inheritance, maternal inheritance of mitochondrial genes. So this is what those look like on a pedigree diagram. Nucleotide repeat expansion in Fragile X mental retardation gene. So here you can see a normal would have just this sequence. A pre-mutation or a carrier would have a bit of an extended sequence. And full mutation would have an expansive extended sequence. And this can be detected by PCR and Southern blot. And by capillary gel electrophoresis. So the normal or pre-mutation and full mutation are found. Detection of Huntington gene, CAG expression mutations by PCR. Human disorders due to mitochondrial mutations are listed below. Kearns-Sayre syndrome, pigmentary retinopathy, chronic progressive external ophthalmoplegia, Leber hereditary optic neuropathy, mitochondrial myopathy, encephalopathy, lactic acidosis, and stroke-like episodes, myoclonic epilepsy with ragged red fibers, deafness, neuropathy, ataxia, retinitis pigmentosa, subacute necrotizing encephalomyopathy with neurogenic muscle weakness, ataxia, rhinitis, and pigmentosa. And here you can see where those are located on the mitochondrial DNA. Homoplasmy, all mitochondria in the cell are the same. Heteroplasmy, some mitochondria are normal and others have mutations. The severity of the disease phenotype depends on the amount of mutant and normal mitochondria present. So mitochondria replicate independent of the cell. So if you have normal and abnormal mitochondria, the rate of replication of each one of those can determine the outcome of the disease. Detection of a mitochondrial point mutation by PCR RFLP. And detection of KSS mitochondrial deletion mutation by Southern blot. Germline mutations, 50 to 100% allele frequency. Mutations discovered by NGS are confirmed by Sanger sequencing. Targeted sequencing panels simultaneously cover multiple known disease-causing gene mutations. Exome sequencing covers 85% of disease-causing mutations. Target exome enrichment is performed by probe hybridization. Sensitivity may be lower than targeted gene panels. Genome sequencing covers coding and non-coding regions. So an example application of NGS. A child was born with apparent autoimmune disease. Surgical and drug interventions were not effective or not sustainable for long-term use. A bone marrow transplant would be effective, but had to be justified due to the age of the child. A genetic component had to be identified. Exome sequencing was performed. A gene mutation on the X chromosome shared in heterozygous state by the mother was found. The mutation was confirmed by Sanger sequencing. The allogenic bone marrow transplant was successful. Genetic testing limitations. Therapeutic targets, except for gene therapy, are phenotypic. So if they're not phenotypically expressed, then we may not be able to accurately therapeutically target them. Non-symptomatic diagnosis, where disease phenotype is not yet expressed, may raise ethical concerns. So just looking for the presence of particular genes when the person doesn't even have symptoms of the disease, it may have some ethical issues. Most disease and normal traits are multi-component systems. Genetic testing complexities, so variable expressivity. A single genetic mutation results in a range of phenotypes. Genetic heterogeneity, the same phenotype results from mutations in different genes, includes diseases with multiple genetic components, and penetrance, the presence of mutation without the predicted phenotype. Molecular genetic testing advantages: rapid definitive testing for known and established genetic variants. DNA analysis has a predictive value for disease risk, diagnosis, and therapeutic intervention. This doesn't mean that it's a good idea to just get whole genome sequenced to see what diseases you may develop in your lifetime. Gene variant databases are growing to address complex changes and multi-component diseases. Gene discovery for inherited diseases was moved from research to clinical applications. And then you can review the summary.